A truck arrives at the Census Bureau carrying 10 million survey forms. Each form was filled out by one person in the city, answering 20,000 questions about their life. Your job: process these forms, find patterns, and map the city's neighborhoods. In single-cell RNA sequencing, each form is one cell. The 20,000 questions are genes. The answers are expression counts. The city is your tissue sample, and the Census Bureau is your bioinformatics pipeline. Welcome to the Census Bureau. First, the inspectors. Some forms are legible, barely any questions answered. These are low-quality cells with few detected genes. Toss them. Some forms are water damaged. The ink is smeared with a specific pattern. In biology, high mitochondrial gene percentage signals a dying cell whose cytoplasm has leaked out. Toss them. Some forms are blank, empty droplets that captured no cell at all. Toss them. The thresholds: minimum 200 genes detected, maximum 5% mitochondrial reads, and dabbled removal for forms that look like two people filled out the same sheet. Quality control is not optional. Garbage in, garbage out. Each neighbourhood submitted a different number of forms. The downtown district sent 50,000. The suburbs sent 5,000. If you just count raw responses, downtown dominates everything, not because their answers are different, but because they sent more forms. Normalization divides each form's counts by the total number of answers on that form, then multiplies by a scale factor. Now a gene expressed at the same level in downtown, and the suburbs gets the same normalized value. Log transformation compresses the dynamic range. Genes expressed at 1000 and genes expressed at 10 are treated more equally. SCTransform does normalization and variance stabilization in one step. 20,000 questions on each form. But most questions got the same answer from everyone. What color is the sky? Blue. These aren't informative. You want the questions where answers actually vary between people: political opinions, dietary preferences, career choices. These are the highly variable genes. Plot each gene's variance against its mean. Genes above the expected variance mean trend are highly variable. Select the top 2,000. These are the features that distinguish cell types from each other. Everything else is noise or housekeeping. 2,000 questions is still too many to work with directly. Principal clerk reads through all 2,000 and notices patterns. Questions about food, commute, and housing all correlate. They're really about lifestyle. Questions about education, salary, and job title correlate. They're about career. The clerk summarizes 2,000 questions into 50 themes, principal components. Each theme captures a pattern across many questions. The first theme explains the most variation, the second explains the next most, and so on. The elbow plot shows where adding more themes stops being useful. Usually 15 to 50 PCs capture most of the meaningful variation. 4 processing steps. Quality control to toss damaged forms. Normalization to make neighborhoods comparable. HVG selection to keep only informative questions. PCA to summarize into key themes. The raw forms are now clean, normalized, and compressed. Next, map the city.